Yes, here we are again. Hi, everyone, and welcome to another Saturday live stream. And yeah, I'm seeing that. Wow, yeah, 33 people have already joined the stream. Okay. And we're going to get started right away. Just give me a second or so. I'm just getting everything organized again. See ya. Exactly. Hello from Norway, from Greece, from Vietnam, from Northern Germany as always, Netherlands, okay, UK, France, yeah, from Upper Austria and from London, the United States, Florida, England, Brazil, Netherlands, and Poland, all from all over the place. And today, look what I got here. We're going to have a look at a couple of, yeah, not only tardigrades, water bears, but I would like to make a mini terrarium because this moss here contains so many tardigrades. Honestly, I've been doing microscopy now for quite a few years now, but I've never seen so many tardigrades in one place before. I'm just going to show you. Okay, and for this reason, I want to basically keep the moss at home so that I can always look at those little water bears. I'm just going to show you here the scope view. Look at this, in one drop of water. I'm going to show you how I extracted those. Look what's going on here. I mean, this is crazy. I mean, here at the top, I think I counted up over 10 tardigrades. Yeah, I'm just going to take the arrow out of here. Let's go up a little bit with the magnification. I'm just going to show you. I mean, look at this movement here. Yeah. Um, yeah, it's absolutely yeah, crazy. I mean, I did not concentrate them. I did not collect them together. But actually, this was just what I found in one drop of water, yeah, from the moss. Yeah. This was honestly, we've got a whole bunch of, this here is a mite, for example, yeah, over here as well. We've got those worms here, nematode worms. This one over here is a tardigrade. Yeah, I think I did a rough count, and I found some rotifers. This looks a little bit like a rotifer over here. Yeah, so lots of stuff. Unbelievable, really. What's going on here? So what I'm gonna do is I'm going to show you a little bit, yeah, how I've extracted those. Right, it's really quite easy, and then I'm going to make this mini terrarium. And then I just realized a couple of minutes ago, got everything prepared, and I just realized a couple of minutes ago, oh my gosh, I think I'm gonna it's such a simple, easy project that I'm probably gonna be finished with a terrarium in 10 minutes or so. So I might be running out of things to talk about. So we'll take, yeah, we'll take our time a little bit, okay? So if you have any questions, it's gonna be like this, that please ask them. My son is going to collect the questions again, especially if they're off topic. But to make life easy for everyone, please write down at Microbehunter or at Oliver, and then it's easier for us to find. Yeah. So very short organizational talk as well. You might have already realized that last week I had to skip the live stream. I was not at home. I was somewhere else. Yeah, because yeah, I had other for private reasons. I had to go somewhere. Well, it was a chess competition of my son, and which lasted a whole weekend, and so I was not at home. Yeah, that's exactly here. How was the chess competition? Yeah, I thought it was fine. My son, yeah, he was not so happy. He only got fourth place, yeah, but but he's enjoying it, yeah. In any case, yeah. So so what I'm gonna do now is before we start making this here, I'm just going to very quickly show you how I extracted those, because yeah, it's pretty simple. I mean, maybe you remember, some of you might remember a couple of months ago I did this the Behrman funnel thing, and complicated. It's actually quite easy, yeah, to extract those. I'm just going to quickly show you that, and then we're gonna yeah, try to make something here, right? By the way, here's a jar, a mini jar. Yeah, that I'll be using. The reason being because I want to keep this moss also at home, so every time I want to collect some samples. Yeah, so and I'll quickly show you. Yeah, I got some rocks here for decoration as well. Some, yeah, and what I've done is the following. I've used those Petri dishes here, and you take a little bit of water, and something that's really important is the following. The type of moss is really important. Do not collect dry moss. Of course, tardigrades are able to survive dryness. We all know that. But I went into the forest. and the moss was fresh and still moist, and this basically means there was quite a bit of biological activity going on. And what you do is you just basically make it even moister, and then what you do is you turn it around, right? And all you do is you kind of knock out all of those tardigrades with a little bit of water, and then what you do is the following, you just squeeze out everything that you've got here, and then you simply wait here like this, and you wait until some of the solid material starts to collect here. You don't want to have too many soil particles in there because they kind of cover everything, right? But if it's very fine stuff that you see here, then it's going to be okay. And then what you do is the following. You take a slide, of course. You have to wait a little bit until all the solid particles sink down, right? And you take a pipette. I'll be using now those disposable ones. You go all the way to the bottom and you kind of take up some of these solid particles here. And what you do is the following. You just wait until all of the solid particles here, soil particles, tardigrades, whatever is in here, you allow it to sink down. Okay, you see, because they're more dense than the water, so it's sinking down, and this concentrates it more. And then when you're ready, you just do something like this, and then you hope for the best. And just to illustrate this a little bit, I'm going to put this also under the microscope. I mean, this was now a little bit agitated. The one slide that I had before, I left it standing for about 10 minutes, so I don't know how many I'm able to find here, but I'm just going to demonstrate this as well. Let me change the scope again here, so you see, look, we still have the old slide. Yeah, now here is the new one that I just prepared. Yeah, and you see there's also quite a bit of movement going on, and of course because of so many soil particles and I don't know, decomposing plant material, lots of the things are actually covered up. So maybe we're just going to go down a little bit, and I'm doing this now without a cover glass, by the way, right? So I'm going to be adding a cover glass as well, and this might spread everything. Yeah, and if you don't see it like that, right, then change the moss. That's the basic, recommendation that I can give you. I've had moss samples there I was not able to find anything. It was pretty much dead, right? And this one is so active, it's unbelievable, right? Yeah. So this that's the thing. Every now and then I'm going to have a look. I'm going to have a little look. What you're commenting over here. Yeah, and what I would like to do now is before I start putting this together, I'm going to drop a cover glass on here because I want to also look at those tardigrades at a high magnification, okay? And right now there is no cover glass on here. I'm just looking at it, the drop as it is. But of course this means no possibility to go high because the distance is too small otherwise. So got here a cover glass. So let me take this out again. I have to change again over to the desk view. Of course, there's always a little bit the danger that when you do stuff like this, that you end up crushing whatever you've got here. So maybe a little bit more water here wouldn't be bad. Yeah, see, it kind of also spreads it apart a little bit more, and this might make it easier to see them. Right, and some of the particles that you have in here, the soil particles and whatnot, right, and they also kind of act a little bit like holding a distance, so that it doesn't completely crush them, right? But you got to be careful because as it evaporates the water, of course, it's going to crush those tardigrades, right? Okay, so I'm going to take out the other slide again, and let's go up here again. Here we go, and you can. It's way too bright. Yeah, and now because essentially everything's spread out more, it's going to be a little bit more difficult to actually find something. It's the same sample, but it's kind of interesting that for whatever reason I'm not able to find anything here. Oh, okay. Okay, where are they? Here is one, for example. Okay, here is one. Let's go up. Okay. They like to really cling, to. Here we go. See, there are also other actually microorganisms around. Yep, and so that's now the 20 times. Let's go up yet further. Yeah, kind of nice to look at. Yeah. So yeah, generally, again, I know I'm kind of repeating myself, but maybe sometimes people are joining in, and that is the moss is critical. So this means if you, I've had very bad results. Sometimes in front of my house there is some moss growing on the concrete wall. It didn't work well. For whatever reason. I think it might be because there are always those moisture fluctuations during the day. It dries out, and the sun shining on it, it dries out. Yeah, sure, those tardigrades are able to survive it, but I think, yeah, when it's continually moist, right, then they're also able to actively reproduce. And then the density is much higher. So what I've done is I've always went into the forest. I collected some moss from the ground or from tree logs. Yeah, and if I kind of feel that it's not completely dried out, okay, then yeah, it's most likely gonna work. Yeah? And here I was very happy that the results are actually quite good. Okay, so and then I would say, I don't know, let's start just putting this together again, and then I don't know, maybe later on we can always continue to watch a few of those. Yeah. So I just also want to say that another thing that what I originally planned to do, I wanted to show you again some fungi and yeah, some mold and mold spores, but I thought, well, actually those tardigrades are always a favorite, right? And people like watching them as well. So yeah, why not? Yeah, have another look here. Yeah, so. For active, okay, I'm just reading this here, yeah, for active tardigrades. So, and what I read as well is that while they are able to withstand dryness quite well, apparently they're not very able to withstand heat. So if you've got basically this sunlight shining on a concrete rock or wall or whatever, then essentially, apparently they don't like that so much. Right, so surviving dryness is fine, but heat apparently is not the best idea. Huh? So let's. I would say, yeah. I'm going to quickly have a quick read here, and then I would say let's start. I will be honest with you. This is going to be the first time. I mean, some of my students have in past years made some of those called mesocosms before. It's a little jar, yeah, and you put soil and plants in it, and you keep it closed. But I myself have not done this yet personally, and so it's gonna be a little bit my, how do you say, all the first trial here. I'm not so, yeah, yeah. It's a question of do they have tails? Yeah, it's not really called a tail. I just want to also mention this a little bit. These are also feet actually with claws. If you count the number of legs, you're going to discover that they have 8 legs. This really confused the scientists back in the day because they could not classify it, right? Because what, 8 legs? Well, then actually some of them actually classified them, well, it must be related to the spiders because spiders have 8 legs. Insects have only 6 legs, right? And they actually have 8 legs, and this really confused some of the researchers at that time. And nowadays, of course, we know that the tardigrades, those water bears, are actually a group distinct from the insects. They're their own category, so to say. So they're not insects. They're not related to the spiders. They're a completely separate group of micro animals. Yeah, and but it kind of shows a little bit how the scientists at that time were kind of wondering a little bit, yeah, what could they actually be? Who are they related to? And the number of legs is actually one of the criteria here. Yeah. So yeah, yeah, there is a recommendation here. Recommended reading the Tardigrades from Hartmut Grieven. Thank you very much. It's in German though. If it is a, how do you say, a historical book which can be downloaded, then of course we can do automatic translations. That's generally something that I recommend. We're kind of lucky here before I start doing this because here the tardigrade is not clinging onto anything, right? Normally it's clinging onto soil particles and then you don't see it so well. Yeah. So we're just going to leave that one over there on here. I'm going to now go back to the desk view. I'm going to add a little drop of water here. So to prevent it from completely drying out. And then let's, I don't know, let's, yeah. Not this one. Let's go this one like this here. Usually add a little bit of water here on the side. Yeah. And so maybe after an hour or so, we're able to have another look at it. So, and now the fun part starts, okay? Because now I have not, we're gonna see how this works out. For me, it's also gonna be the first time that I'll try this. I collected some moss. The moss was moist, and I used a spray water bottle to keep it moist. That's way too much, right? I've got some gravel over here, collected from our playground, and I rinsed it. So it's simply to get, everything, the larger particles removed. This will be the jar that I'll be using. Okay, and I have over here some commercial garden soil. So this is directly from the shop. Some decorative rocks. Maybe not. I don't know. I was kind of thinking, should I include this one as well here? I don't know. It's a little bit too big. I just discovered. But what I'm probably going to do, and I just want to say this here, is I'm probably not going to close off this jar, okay? I've seen already in the past that students who have made this, and they kept it closed. It's called a mesocosm, and the idea is that it's like a self-contained little mini ecosystem. So during the day, photosynthesis happens, oxygen kind of builds up, and during the night, CO2 is being produced by decomposers, bacteria, and the insects that are in here. So it's supposed to be kind of like a closed little ecosystem with only sun able to enter. However, these are very unstable, and for this reason, many of them started to mold. So I don't know. Yeah, so this is maybe one of the reasons why I might keep it open, or at least regularly air it a little bit because especially small terraria are a little bit unstable. Okay, so the first thing that we need is we need to add a layer of gravel here because we do not want to have any standing water. That's because otherwise it's going to go anaerobic, and if things become anaerobic, then you have a real problem. Because then you have anaerobic bacteria starting to grow, and so that's number one. I don't know how much. You see, it's a little bit moist because I was kind of So and this is kind of a little bit the way that we're gonna do it in order to allow any excess water to drain. Normally, if you're using a flower pot for flowers, then of course there are holes on the bottom to prevent standing water, so any excess water is able to run through. And I would like to add a layer of soil, but the problem with the layer of soil is then the soil is going to go into the gravel, right? I hope that this is going to be enough. So what I've done is look what I've got here. This is kind of a net for insects, against insects for the window. And I simply cut out a round piece here, got more of this stuff here, right? It's not very, how do you say, solid, right? I mean, there should be probably more solid netting, but I guess it's going to be okay. Yeah. So this one goes in here to prevent the soil from actually falling through. As a kind of worried is I'm a little bit worried because actually I am going to be finished in a minute or so. So and then I'm going to add a layer of soil, and the soil is important not only because it contains, of course, lots of bacteria which can be a food source, but I've seen that there are also even some insects in here, probably some other organisms as well, and we're just going to try it like that. We're going to add a layer of soil. I need to get a vacuum cleaner too. I don't know. Yeah, see, some of the soil actually is falling through here. Yeah, it's an experiment. So I guess it'll be enough. Let's add a little bit of I don't know. Where's this? Here it is. Okay. So, and I don't know, what else do we have? We're gonna place a couple of rocks in here, maybe a rock or two. Oop. Just, I mean, it's decoration really. Yeah. And I don't know if this is actually really very meaningful or not, but it's a little bit also a little decoration that we have. And I'm going to be adding some moss now. Okay. I carefully, as you can see, I carefully peeled it off from a tree log to keep it together, more or less. And with time, it's anyway going to grow and it's going to start to take on a new shape. And yeah, some of those smaller pieces are quite nice because I can easily take them out later on and kind of extract some microbes if I want to make some other videos again, and tardigrades and so on. And maybe, yeah, look, I collected way too much of this. Oh, this one is actually a much bigger one here. Here's a smaller one. I don't know. I don't know. Is this already finished? What else do I need? I need this here. Where is it? Here. Some months ago, this is what I found here. A dry piece of wood with some lichens on it. I also had some tardigrades on here. I'm going to also add this in here. And it's going to hopefully soak up some of the moisture. And yeah. Okay. Okay, I'm reading every now and then. Yeah. Something over here. Okay, and I don't know. I just realized, should I try to put this here in as well or not? You know what? Why not? Somewhere maybe at the side a little bit. So how am I supposed to put that in here? You know what I'm gonna do? That's why you've got these cool tools called a spatula. I'm just gonna make a little hole here. And it's going to find the roots on it anyway, going to find their own. Or maybe I'm going to add a little bit more. Look. On the side a little bit. Yeah. I guess that's enough. See, now I can't close it anyway. Yeah. Okay. Most likely, I know I have seen a couple of insects here as well, and yeah. So it could quite well be that there's some kind of insects that are kind of So what I'm gonna do, so that's basically how it looks like. I'm gonna show you. I don't know here. It does look kind of nice. It's too small, I just realized. Yeah, I mean, yeah. But at least, maybe, you know what I'm gonna do? I'm probably gonna take out this plant again because I can't close it. I'm just going to put it back into the main pot. I'm just going to leave this here like that. And I'm just gonna put a lid on top, and I'm going to see how this develops. And now I have a source of tardigrades and maybe other insects as well, okay? So let's see again how the other tardigrades are still doing. And then I'm going to do a little bit of soil microscopy as well, because I also want to show you how it's possible to extract some of the things from soil. Let me scope view. Here it is. It's way too bright. You see, that's also quite a nice little demonstration that I put the cover glass on top. Yeah, and now all of a sudden, where are they? And I don't see any movement anymore, right? And what I think happened as well, if you don't use enough water, then their movement is so limited that you cannot see them as easily anymore. And sometimes they get crushed. Yeah, so that's also one of the reasons why I sometimes like to look at those without a cover glass, or we can use those concave slides so that essentially there's a little bit more movement possible. But this one over here, for example, that's the conidia from a fungus. That's most likely from a fungus. Yeah. But I think we can see quite nicely that the cover glass has kind of limited the movement quite a bit. And I don't know where I put the other slide. Oh, here it is. In contrast, the other slide. Yeah. You still see a lot of movement here. If I put a drop of cover glass on top, then again it's going to be so squeezed that it's probably you won't be able to see the movement anymore. Yeah. Okay, there's some discussion going on here a little bit about where to put the mesh. I suppose, now I have not read all of the comments, I suppose that you put some of the mesh over some plants to keep the slugs and other snails out. Okay. And moss leaf would be interesting to look at. Yes, we're going to do that as well. Okay, so yeah, but I just wanted to show you over here again why I did not originally put on a cover glass, because it kind of spreads out everything, so the density is going to be, of course, lower. But at the same time, if you don't use enough water, it's kind of going to be crushed. You know what I'm going to do? I'm going to add a little bit more water here to prevent evaporation. Okay. Yeah, here we go. Yeah, but again, so what you can do as well, I've tried this, it takes a lot of patience, is to take a pipette and kind of pick out the individual organisms and tardigrades and transferring them. But this is quite a bit of a, yeah, takes a very calm hand. How much magnification can you go without a cover glass? It depends, of course, a little bit, but what I'm doing here is I'm not going above, yeah, it's 20 times. And no cover glass. But you see, it doesn't make a lot of sense, because the sample is so thick and the depth of field is so low that it's very blurry. So I would not go much higher than than the 10 times magnification. Yeah, and now I've got approximately a centimeter distance, and so that's not a problem. Yeah, of course you have to be a little bit careful that the water doesn't spill over, right? Yeah, and this one here, so with the 4 times. I have to flip out the condenser to get the full view. Yeah. You get the idea. Okay, so what I'm going to do now is, because of request, let's have a look at the moss itself. But before I do that, I do want to clean up the whole table a little bit. And we're going to see how this actually develops, this jar, because occasionally what happens is that some of the, either the moss starts to grow really quickly, but then sometimes also other plants start to grow, so you cannot really predict this all the time. Fungi can start to grow, that's always a problem. So some people are actually recommending that you put some insects in there, like springtails, which are then eating the fungus, and so that's also, AP exams, that's advanced placement, that's in the United States, these are final examinations. AP exams it's referred to as advanced placement, and these are some final exams for university qualification. Would it be an idea to sprinkle a few sand particles as a distance holder? That is possible. However, I would say in many cases it's probably not always necessary, because very often there are plenty of sand particles and already in here. What you can do is that, and I have actually shown this last time a few weeks ago, the so-called in a video. I don't have it here right now, but there are slides with a concave slide which allow, which have a little bit more depth. Or what you can do as well is you can, and I might as well show that to you. Why not? Because there's a question here. Is you can also do the following. Look, I'm going to take again some of the. Let's do this here again. I'm going to show you here. You have to wait again until everything kind of collects on the bottom. kind of collect all of this here. You allow the whole thing to sink down again. Put it on here. And what I'm going to do now is the following. I'm going to put a cover glass over here on the side and another one across. I think is this one or two? Okay, that's one. Good. And another one, I'm just going to put like so. It's kind of a little bit diagonally, right? So this means that some organisms, that are larger have more space over here. The smaller ones over here, but you can actually see that over here. The water is not even completely spread. So this means that the grains and the sand grains and the particles kind of are also holding up this part here. But we're going to have a look now. Let's have a look of how well this actually worked. Let me remove the... So where is this? Here we go. Let's get an overview again. So this is where it's narrow. Oh, look, there are some other interesting organisms here as well. What is that? And over here, it's a little bit, further away. And here is the other cover glass. Yeah, but most of them are over here, and you see that there is still a little bit of movement possible, or quite a bit, because there are so many other particles here. Yeah, here's one. Here's another one. 1, 2. Honestly, this is one of the most, 3. This is one of the most successful moss samples that I've ever looked at. Yep. Normally you do have to look quite a bit, but here there are quite a few of them. Here's another one. You get the idea. So you can experiment around a little bit with kind of keeping the proper distance. They're adorable. Adorable. Our tardigrade's adorable. Look, I'm going to show you something here. Maybe some of you already know this. A 3D print. I don't know, I found this online here. Because actually tardigrades look pretty creepy looking. Now you don't see it. But actually, concerning the anatomy, these models are quite nice. Look, these are 3D printed models of tardigrades. You see the 8 legs, a mouth over here. Look pretty scary, and they actually sometimes they cannibalize each other. They eat each other. So just to show you a little bit how it looks 3-dimensionally. And I don't know, there are plenty of these different species of tardigrades. What they feed on bacteria, debris, and so on. So, just wanted to show this to you as well, as a little bit as an orientation. Let's have a look again at the scope view over here. And there are some other interesting critters. What is this here? Looks a little bit, that's a rotifer. Lacrimaria has this one protozoan with an extremely flexible long neck. Okay, you get the idea. We'll keep that here, and I'll just put a little piece of moss now under the microscope as well, because this has been a request. Okay. So... I need to grab a slide. Some people have been asking me what do I do with the slide after I've used them, and I'm just telling you right away I'm cleaning them. So this is again a little bit of a difficult thing because I need to get a... you cannot see that. Because here we go. This is very thick. Okay, maybe this is better here. Look. Okay, this is very thick. I'm going to take a small water sample. Okay, cover glass. You see, way too thick. So let's add a little bit more water. It's not a big problem really, but yeah. So let's see how this looks like. Yeah, you see, way too big as a magnification. And what do we have here? Yeah. There's also some kind of a tardigrade hiding here between the leaves. Look, difficult to see, but over here, have a look what's going on here. They like to cling to the leaves, and then of course what you have to do is you kind of have to rinse them out or wash them out somehow. I don't know, they have disappeared. Let's go down to with lower magnification again. Yeah, but here it's a little difficult to see if there's any. Okay. So. Here we go. Yeah, though, if we actually zoom in a little bit more, we can actually see that the individual cells, the leaves should be actually quite thin. Maybe they're only one cell layer thick. Here we go. That's nice. Yeah, it's not completely flat. So let's go up 60 times. Okay. Yeah, the cells of the moss are fairly small, I think. Yeah. But we can see that evidently the moss is one cell layer thick. Here we go as well. Yeah, so sometimes we actually see much more. Yeah, the individual chloroplasts, those tiny little dots that you see in the cells, right? These are chloroplasts. Yeah, for photosynthesis. And what we actually should be able to see as well is the following: is if we are now very patient and if we wait maybe for, I don't know, for an hour or so, we should actually see it doing photosynthesis, and then you see oxygen bubbles starting to grow. Yeah, and you can do also a time lapse with that. Yeah, so if we happen to find a bubble somewhere, then we could actually also watch it grow a little bit because that's photosynthesis happening. Sometimes those bubbles appear at the on the bottom side of the here, where it's kind of cut off. So sometimes you can actually see them appear here out of the stem as well. But actually thinking about that just a second, I think I made now a biological mistake, because mosses are not vascular plants. So maybe they don't collect over there the gases. Yeah. So let's go down again a little bit with the let's here, and let's see if there's something maybe here on the other one. Maybe there's still a couple of I don't know if tardigrades cling to it. I already washed out that moss before, so of course many of the tardigrades have already been washed away. Oh, you see, there's a bubble here, or two. What we could do is we could patiently wait and see if it actually does grow. What's the stem? That's an interesting way. I'd have to cut it. Yeah. To see that. Do you use a dummy battery when filming with you? Yes. Actually, I don't use otherwise I might run out of batteries. So what I've got is I've got it the Canon camera I've connected over a power supply. Yes. Okay. There are some off-topic questions. Yeah. Basically, many water plants are very thin. because they do not have to worry, or also moss, because they do not have to worry about dehydration. And why do they have to not worry about dehydration? Because they are always surrounded anyway by water. So yeah, so yeah, that's basically the moss. Okay, let's have a look again at the. You know what I have to do? Actually, I should. No. The microscope actually allows me, and I was simply too lazy to do that, allows me to actually program it in such a way that every time when I change magnification, that I do not have to manually change the light intensity. But yeah, here there are two of them next to each other. That's nice. Yeah. So what time is it? Okay, yeah, I'm always checking a little bit. Yeah. I mean, what I also have prepared, and maybe we're just going to watch them a little bit for a couple of minutes more. Then I would like to show you some other things that I've prepared here. The fungus. Yeah. They're always surrounded by water. Beginning of the stream. Don't use dried moss. Yes, that's the thing. Moss being a non-vascular plant. Basically will take up the water all over its surface. You see, normally vascular plants will only take up water over the roots, right? But mosses don't have true leaves and true roots and true stems. It's referred to as a so-called thallus in biology, and therefore they are limited in the areas where they can actually grow. Yeah. Okay. Why aren't aquatic plants thick? I mean, there are some water plants that are fairly thick, indeed. But many water plants, like Elodia, for example, are quite thin. Because they do not, because thick leaves with a thick waxy cuticle and so on are actually there to prevent water loss in terrestrial plants. And this water loss is not a problem obviously with water plants because they're always surrounded. And for this reason, it is not necessary for them to have those thick leaves. But I mean, there are some water plants which do have that. Yeah, yeah. So let's play around a little bit. Okay, I got it. It's here. Here we go. Yeah, I have to play around a little bit with the filter so you can change the colors, as you can see that. Huh? Okay, here we go. So yeah, these they look nice. They look cute. They're interesting to observe. I would say probably difficult to make a permanent slide. Yeah. By the way, many of these micro animals, and these are true animals. These are actually, per definition, not microorganisms. The question is why? What's the difference? The reason is because these tardigrades, these water bears, are multicellular, made of many cells. Multicellular animals also have cells that are specialized. And if you've got specialized cells, then you're talking about a multicellular animal. And not only that, those tardigrades are so-called eutelic. And eutely means that they have a fixed number of cells. Very different to us humans. We humans have a different number of cells. But within a species, that is, of course, those tardigrades have a fixed number of cells. And actually, in some organisms, I think in some nematode worms, they have studied it so much they're even able to trace exactly from the fertilized egg cell onward the development of every cell in their body. I think it's kind of fascinating. The question is now, how did they find that out, really? That how do you count the cells of an organism like that? And I think I've read somewhere that the early scientists, they have been doing some kind of a staining techniques. And they were kind of staining the cells or the nuclei, and that's basically how they discovered that by counting the cells, that essentially they have the same number of each one of them has the same number of cells. Yeah? So, yeah, occasionally I'm reading again. Okay. Yeah. And I don't know, some time ago I actually made a a video about this because every time when I look at those critters under the microscope, sometimes I'm getting a little bit philosophical, right? And the question is now, how did they look cute, those tardigrades, but actually are they intelligent or not? I mean, I'm getting these questions, and after a bit of research, I have to say, no, they're not very intelligent. They're actually pretty, under quotation marks, dumb. Because they do not have a truly developed brain. So they have so-called ganglia, and the ganglia are connections of nerve cells, and they do not have to be intelligent because for their environment, it's fine what they're doing, right? So they don't seem to be using it to backfeed much. Yeah, that's correct, but I have seen actually some of them to use them to mainly to cling to a surface. So they're using the front 6 feet to kind of move ahead, and the last 2 pair of feet for clinging. Yeah. Is it possible to illuminate with reflected light? You won't see a lot. And the reason is I don't have a flashlight now with me. I mean, I can show it to you under my stereo microscope. But the thing is the following is because they're fairly transparent, and for this reason, the contrast is really bad with reflected light. Okay, but because you're asking, I do have a stereo microscope, and I'm just gonna show it to you also under the stereo microscope. But please don't be disappointed. Okay, don't be disappointed because you're not gonna be seeing a lot. Need to prepare this, and let me quickly get the stereo. I hope that everything. Why is there no? It's interesting. There should be actually the stereo stage view as well. Let me quickly see if I'm able to get that started. I don't know. Yeah, actually, there should be I should be able to see the stage of the stereo microscope as well, and for whatever reason, it's not showing that. Oh, actually, it is. No, is it or is it not? No, that's not the right one. No, it's kind of a pity. Here we go. Now it's here. Haha, I'm happy. Okay, so let's do the following. Let's take this here, and I'm going to show it to you now with the light coming from the top. So that is now the specimen that we had just had. I need to focus. Right? And that's how it looks like. Yeah, with a stereo microscope, and magnification not high enough. Let's see. I mean, you might be able to see a little bit of movement, but honestly, this is not. Yeah, it's not the best, right? The magnification is simply not. Let's see. I mean, it's good to get some kind of an overview, and you are able to see a little bit of movement, yeah. But the contrast is pretty miserable, yeah, because it's you need to have real objects that are really opaque, and for transparent objects, it's not really suited. You know what? Just for trying it, I'm going to put a darker background here. If this makes some kind of a difference. Yeah, also not. Yeah. So yeah, let's put the moss directly under the microscope, because the moss itself is again a little bit it's more opaque. And this is how the moss itself looks like. But not surprisingly, to be able to see individual tardigrades here, that's not going to be so easy. Yeah. Yeah, so yes, that's has been the request. Yeah, so that's the lowest magnification that I can obtain now, right? You see that's you know what? This is very covered with water, so I'm going to take another one that's a little bit more dry. I don't know if this is. It's also covered. Yeah. And Honestly, I have not managed yet to directly find tardigrades, yeah, but under the stereo microscope like this. Yeah. Löffler solution. You guys want me to actually do some staining? You actually want me to do some staining? This might kill them, but you know what? They might die anyway on the slide. So let's do some staining. Not Löffler's solution. I will tell you why not. Because Löffler's solution contains alcohol. And alcohol is a problem, and it contains potassium hydroxide, and that is a problem. And for this reason, I'm probably not going to add Löffler's, but maybe we can try to add some crystal violet, or maybe saffron in water. It's got to be a water-based. I mean, crystal violet is a very strong stain. Okay, you know what? Let's just try it. Okay, let me get my stains. Just a second. Wrong box. Here we go. Let's clean up everything here. So let's see what I've got here. I'm just going to show you the box as well. So I've got, what do I have here? Iodine, nope. Acetocarmine for DNA staining. Very acidic, nope, it's gonna kill him, definitely. Löffler contains, that's methylene blue, but it contains also potassium hydroxide, and it contains alcohol for DNA staining. It's definitely gonna kill him. This one here, fuchsine, that's an aqueous. We could try that. I don't know how well that works, but I want to try saffronin. Yeah, I don't know if that stains. And this one is a very powerful one. It's crystal violet, but it's almost a little bit too powerful. So what I need to do is I need to really dilute this down a little bit. Because otherwise, if I don't dilute it, it's going to be way too strong. But generally, it's a very good stain because it's a pretty strong stain. But we have to dilute it down a little bit, and then honestly, this is going to be the first time that I'm going to be staining tardigrades. Honestly, so we're going to do a small amount here. Let's do the following. So here we go. This is what we call live staining, and the rule in live staining is not adding too much. Yeah, not too much, otherwise it's going to kill them. So we're going to go back to I'm going to take a little sample here. And I don't know if this is enough or not. Okay, I really have no idea. I really don't know. And the scope view, here is one. It's fairly to the side of let's take the arrow away. Now, of course, there is now the danger. If I now add the stain, what's going to happen is it's going to flush it away. We're going to see. Okay, you can also check what I'm doing up there. So here we see it's already kind of, yeah. I'm going to switch away so this is now bright field, okay? You see that some things are already stained blue, whatever that is. Okay. So some particles are now staining blue already. Let's now try to find some kind of a tardigrade here somewhere. This guy over here, I don't know. I don't know if... see, over here, those particles are blue. Maybe this one is also going to turn blue. You see the surrounding particles are turning blue. So this means the stain must have already reached it. Let's see what happens. Let's see what happens. Yeah, there is vital stains are used for living organisms. The vital stain kit's very cheap. Yep. But you see, we can already see that the surrounding, yeah, there are some surrounding things that are already turning a little bit bluish. And we're going to see if this this tardigrade also will take up the stain somehow. Or maybe I need, I don't know, maybe a little bit more. I don't know. So what I'm going to do now is I'm going to be brave. And I'm going to add a little bit more. Yeah. Rose Bengal. Ah, interesting. Yeah. Ah, okay. No calculations, pure counting. Ah, thank you very much for the info. I have the book in front of me. Epidermis, 446 cells. Stomach lining, 19 cells. Brain, 39 cells. These are the tissues I can translate. Oh, thank you very much for the info. So this means you've got indeed some info about the number of cells. Could it be? Look a little bit. Is it? And I don't know if you remember, I tried to feed some of my Ailosoma worms some stained yeast cells and so on, which did not quite work. But I might actually try that with those tardigrades as well. I'm just going to isolate a few of those, and then I've tried to feed them some yeast cells, some stained ones, and then hope that you're able to see them actually take up the blue cells. The terrarium, yes, I put a layer of soil into the terrarium as well, simply to, how do you say, keep the moisture a little bit better. Okay. I mean, maybe it does turn a little bit bluish, I don't know. So what I've done now is I've added a little bit more of this crystal violet stain into the water. And I'm going to now add a little bit more here on top. Into the slide. Hop. Whee. This was, maybe you could actually see how the stain flushed across. Yeah. Now I have to, of course, find them again. Maybe some of them were kind of flushed away. Let's go down with the magnification yet more. There's a nematode worm over here. Yeah, as the stain went in here. Thank you. So your questions are here. Where are the tardigrades now? They were kind of flushed away, and maybe they can be found somewhere over here. Let me check. But then again, I'm at a very low magnification, so maybe they are difficult to see because of that. So what I am going to do therefore now is the following. I'm going to try something different. I still want to stain them. I'm going to take the drop that I have over here. So I'm going to remove this. I'm going to do something, and I'm going to take this one over here. So that's the one from before. Okay. So, and I'm now going to directly add into this drop a little bit of this stain. Let's see how this actually works and whether they're able to survive it. Okay, there are two of them here. I need to focus a little bit. So this one here is now touching the stain. I mean, the other one doesn't seem to like it a lot. Let's see if it's able to survive this staining. So 20 times. So that's going to be interesting. It's hiding now here behind this grain. It's difficult to see. Ah, yeah, there is one over here, which might be a little bit better. But here this is a little bit risky because, of course, the objective is very close now to the drop of water. But I have to tell you the following. That's also quite interesting. A little. It does not seem to actually stain very well. Because you actually see all of those many other particles here are stained really dark. But normally what happens is that cells that stain well will accumulate the pigment and will actually turn darker than the surrounding, and they don't do that. Yeah, this kind of shows a little bit that not all stains are suitable. Yeah. Yeah. So rose bengal, I don't have that stain, unfortunately. What causes the tardigrades to go into the ton state? The ton state is a state where they're able to survive dryness. What I have already done before is if you slowly dry those tardigrades, you allow the water to evaporate, and then they will kind of pull together and yeah, become smaller. They don't look like a tardigrade at all. And then when you add water again, over the course of maybe about half an hour, it takes much longer than actually, they're kind of for coming back to life. They will actually then expand. They will slowly start moving again. I was actually able to observe this once, and looks quite interesting. But I think it's also like this, that you are not allowed to dry them too quickly. And look at this. This worm over here, this nematode worm, is not feeling so happy. You see, it's kind of moving already in a very uncontrolled way. And we can actually now see that the tardigrade is starting to become more and more bluish. Especially if we turn up the light. Yeah, so the cells are now actually starting to absorb it. Now we can actually see it much better. Yep. Yes, now we can actually see it's much more purple. Their cell walls are too thick. Let's put it this way. Ever smaller Jesse was saying their cell walls are too thick. I have to correct you. These are animals, they do not have a cell wall, but I get what you mean. So the cell surface, so to say, or maybe they have a skin made of chitin, maybe. I'm not so sure. Maybe someone can research that. So that means it indeed might be the case that the pigment is not able to penetrate into the cell. Yeah. They're fluorescent dyes. It is like this that in the case of fluorescent microscopy, that's actually quite interesting. It depends how specific they are. There are so-called fluorescent antibodies, and what they do is they can be used to highly specifically stain certain parts of those tardigrades or of cells. And then there might be tripolarized light. Actually, the problem is the following. I do have polarization, but currently it's only in association with DIC that I have. So it's not cross-polarized light here. Okay. Cross-section of a moss stem, that's going to be a little bit of a challenge. I don't know if I'm able to do that, but why not? I need just a second. I need to find something here, because this is part of this format. If you request me that I do something, then I will try to do it, even if it is not successful. Okay, so that was not a staining. Okay, luckily it did not touch that. You see how some of the parts really stain blue? Huh. Yeah, the tardigrade slowly is. So what I'm gonna do now is because due to popular request, let's try to stain now. Not stain. Let's try to do a cross section of the of a moss, and I'm going to be using my traditional way. But honestly, people, I don't know if it's gonna be successful. Okay, so that's always Can I do a one-handed cartwheel? Nope, I cannot do that. Honestly, I gotta be careful about sports, because sometimes I hurt myself. A couple of years ago, I tried some ice skating, some things, and then that's why I'm not doing any dangerous things. Even if you request them from me, I'll only do those things that I can do on my desk here. Ultraviolet light, honestly, I don't have 365. I mean, I do have a UV flashlight somewhere, but I think as so often, ultraviolet light has the problem that it does DNA damage. And so I can imagine that essentially UV light might actually also ultimately kill them. So I'm going to do now you don't see what I'm doing. So I'm going to do the following. Let's have a look at the desk again. And honestly, I have doubts whether I'm actually able, to get a proper cross-section here. Really. Because it's so thin. Yeah, so let's try it. Okay. So, and I'm going to show you how I do that. I mean, I've got this... Doing a cross-section of this plant here is way easier, but sometimes I do want to be challenged a little bit. So what I'm going to do now is the following. I'm going to switch over now. I'm going to take the microphone along a little bit, so maybe it's going to be a little bit softer. And even if I may disappear, you should be seeing the stereo microscope here. And what I'm going to do now is the following. I'm going to put, and you can see what I'm doing up there. Just a second, like this. So I put one on top. Let's do it like this. It's way better, because I would like to get a cross section here, and I have somehow. This is difficult. With a pine needle, with a pine leaf, it's probably possible, but this one is gonna be super difficult. So, and then it's probably no. This is not gonna work. You see this? So that's it. That should be the cross section. Let's try another one. This is super difficult. See? This is super difficult. I don't know. No, I was not able to cut it. But you know, let's have a look here. Yeah. So crystal violets on the leaf, that's something I can easily try. But you know what I'm going to do is I'm just going to try to have a look at this here. And let's see if I'm able to see anything here. So, and I'm going to change over now to the scope view again. It's way too bright. I have to find it. That's the one here. Let's zoom in a bit. No, it's still too thick. Not good. Not happy with this. Yeah, you see it's too dark here. Yeah, so honestly, I think, what time is it? Yeah, I think I'm going to try this and maybe I'm able to report back. But getting a cross section here is quite difficult. But somebody requested, why not try to stain them? And that is indeed something that's easily done. And here it is. This was this one over here. So let's try that. Here we go. And I don't know. I'm just going to do the following. Yeah. Let's put this away. And I'm going to take some of this diluted. And now I am going to put a cover glass on top, because this one has to be pressed flat. See, this is not good. It's still too thick. But maybe there are some parts here. Let me quickly check. Oh yeah, interesting. I don't know. Let's see how this works. See, that is something a little bit more interesting. So that's now basically stained with crystal violet. And you were asking for stomates? Let's see, I don't think that they actually have stomates. So let's open this up. Yep. Not able to see. So, but the cells did take on the stain. Yeah. So I'm just reading. Yeah. Don't use UV light on your skin. However, there is one interesting little experiment that one might try to do. It's called autofluorescence. Essentially, if you're blasting a blue light or UV light of a certain wavelengths on green chloroplasts or algae, sometimes they will actually start to glow in red. But honestly, I think this moss here is, as you can see, it does pick up the stain a little bit. Yeah. Yeah, I have to tell you, it does pick up the stain, but it doesn't really have a lot of, how do you say this, differentiating characteristics. But maybe one of the things one could try to do is with those, and I've tried this as well, is you add a little bit of this stain on one side of the plant, and you can actually see how it actually travels up the plant and how it starts to stain the different parts. Yeah. So you know what time is it? Yeah, in a couple of minutes, I've got some of your questions still here. What I need to do, what I want to do first is, just a second, let's have another quick look at how the tardigrades are doing. That's again the stain sample. You see that a lot of the background blue stain has disappeared as the stain has been taken up. And here, yeah. You see over here lots of whatever particles that have taken up the stain, but the tardigrades here, they have survived that treatment, but they are not really particularly interested in taking it up. Okay. Can you make them photosynthesize under the microscope to see generating oxygen? Yes, I could do that, but this takes a long time. I've done this also with some algae before, and you can actually see some oxygen bubbles then starting to grow. Yeah. Yeah, so here we go. Here's another one. Here's some more. Yeah, you get the idea. Okay. What time? Yeah, I'll quickly have a look. Maybe I already answered some of those questions before. Yeah. Let's quickly have a look here. Yeah, how was the chess competition? Depends who you ask. He's never satisfied with the results. Moss on lichens from a shade seem to be from the shaded side of trees seem to be best out of the direct sunlight and after recent rainfall when it's still moist. Yes, I've also seen that essentially it does take some time for those tardigrades to come back to life if it was very dry. And indeed, I've been very successful finding those tardigrades from moss that was not directly exposed to sunlight. Okay, looks like I finally have some use for the stuff growing on my shed. Yes, okay, it's yeah, that's an interesting one. Okay, thank you for the recommendation. Okay, they can use them to cling onto things when they need, but to separate. Yeah, okay, it's not a question. I understand that tardigrades need to keep a film of water on them even when they're on moss. Is this so? Why do they seem to prefer living in moss instead of in water? Let's put it this way. Yes, it is correct that many small organisms prefer to have a layer or thin film of water on them. And as a matter of fact, they are made of mostly water. I mean, we humans, for example, are 70% water. And if there were not a thin layer of water on them, then they would, of course, evaporate. Then why do they prefer living on moss instead of water? And this is almost a philosophical question, which goes deep into evolutionary theory. Basically, what let me reformulate this. In other words, if you say, why do they seem to prefer, then you're essentially to translate this is what's the evolutionary advantage of living on moss instead of in water. And here I have to say, they're adapted to live on there, and for this reason, it works for them, and it's good enough for them to survive. It kind of sounds weird if I say it like that, but from evolutionary theory, that's actually the way it is. I mean, it works for them, and therefore, it's good enough for them, and they're successful, right? And they they are adapted to live in that ecological niche, right? The question is, are theoretically, why don't, in other words, I could say is why do we not all live in water then? I mean, even as humans. Well, and the answer is because we have adapted to life on dry ground, and our bodies are made that way that we're able to survive that way, right? And it's the same also with other organisms. Yeah. So moss under the stereo, I've done that. What type of ISO do you use when taking pictures under the microscope? Okay, so well, I'll be honest with you. What I'm doing, I just show it to you. I don't know even the ISO setting, but generally I use very low ISOs because I generally have enough light anyway. So I just want to show this to you now. I can change the exposure by turning a little wheel on the camera, which is the exposure time. Yeah, so that's one thing that I can do. And or I can actually change the brightness directly. Yeah, and I think I've set everything to ISO 100. Yeah, and what I'm doing is right now by turning this little adjustment wheel, it's I'm changing the exposure time. Yeah. So where's the desk view? Would it be an idea to sprinkle a few sand particles? Yes, that would indeed be possible. However, I think that actually sand particles, it depends a little bit on their size. You'd have to make sure that they're fairly small because larger sand particles are actually, quite big. Most of them are actually too big. Yeah. Yes, I'm using a dummy battery. It's directly connected to a power supply. Reflected light, unfortunately, I don't have, I'd need a strong flashlight right now, and then it would work with the low powers, like 4 times and 110 times. However, I think it might not work quite well because they're transparent. Yeah? Tardigrades must choose dense types of moss that hold a lot of moisture so they remain moist themselves by contact. Yes, I can imagine. Having said that, there are probably many thousand species of different tardigrades, and I'm quite sure that different ones are adapted to different moisture content. So even that is quite well possible that there is not, one type, one size fits all, so to say. So let's have a quick look again. Over here into this view over here. Let's see if we're able to find a few more. Well, that's a mite or something like that. Nematode worms, plenty of worms. Now I have to find the tardigrades if they're still alive. Let's see. Because sometimes there seems to be now less movement than before. Actually, way less movement than before. And the crystal violet that I added, is much less right now. And I think, it must have been kind of absorbed. That's also interesting, and or broken down. But look, it looks pretty dead. Look, there is a gigantic mite over there. This one actually, this one we might be able to see with reflected light. I don't know if I'm able to find this. So let's see how this looks like under the stereo. Here it is. This guy's trying to escape. You can actually see the blue color here. Is this even a mite? I don't know. Gotta count the legs and it's too dark to see. Looks actually, simply from the shape, it does not look like a mite, but see, that's now reflected light, and still not a lot to see here. Does it have 8 legs? 1, 2, I don't know. How many legs? Count the legs. 6 legs, and it's an insect. It looks a little bit like a tiny beetle. Actually, it does look a lot more like a beetle. No, but I think it has 8 legs. Yeah. Okay, it doesn't matter. Yeah. Yeah. That might eat everything, you know what? Wouldn't surprise me either. Wouldn't surprise me either. Yeah. And it's too difficult for me to flip it around as well. Let's have a look if I'm able to. No, it's impossible. Look at how big this is. Yeah. Impossible to flip around. Yeah, I think I'm just gonna, yeah. And I think even if I were to be able to flip it around, I might not be able to see the legs because it's so dark. What are the differences between mites and ticks? Well, they're all both belong to the arachnids. They're all related to spiders. And ticks actually are mites. Yeah, ticks are blood-sucking mites. Right? And how is that micromanipulator coming along? Not at all. I've been planning to build one, yeah, and but honestly, I got a little bit stuck with that. Okay, so what I'm gonna do now is I'm just going to have a quick look at some of the comments that you've written in case there are some questions that are overlooked. Yeah. Yeah. Can you make them full? Yeah, that's basically yes, indeed. If you check, you gotta check somewhere in this. For those of you who don't know, I've got 2 channels, right? The one that you're watching right now is called Microbehunter Microscopy, where I just recently started to put back in the live streams, and the main channel is called Microbehunter, and I'm doing more like microbe explainers right now. So I tried to clean up the channels a little bit. So in the other channel, you might still be able, or you're most certainly able to find quite a few videos where I've put algae and other plants under the microscope, and you can actually see the bubbles growing. Yeah, so by photosynthesis. Yeah? I think there are no further questions here. Okay. Yep. I hope I'm not overlooking anything. Yeah. So here it's going again. Yeah, I think people, I'm going to do the following. I'm going to call it a day. What's the best way to? What's the best way to kill a tardigrade in order to observe the internal structures? I don't know. It depends if you want to make. If you want. Normally, what you would probably do is you can try to work a little bit with alcohol. Alcohol has the problem, however, that it dehydrates and it causes the cells to shrink. You might also try maybe some acid, some pH changes maybe they don't like. So I would say, not that I know of any official way, but I'm quite sure that there are some that are documented and that are kind of standard in research. Whatever, if you kill them, then what's important, however, is that the substance is added that they do not introduce any artifacts. So, for example, shrinkage of the cells or so on. Yeah. So I have to tell you, I'm a little bit tired already. After 1.5 hours, and I'm quite sure that many of you are as well. And for this reason, I'm going to just call it a day again. And I'm going to think of something else for next week. And we're going to see how the little, because that's how we started off with, how the little terrarium is going to work. right? And maybe if the little piece of wood here with the lichens remains moist, then of course also organisms that are on here might come back to life as well. But I think I'm just going to call it a day for today. I wish you all the best. Yeah, and I think yeah, happy microbe hunting. I think that's what I usually always say, and hope to see you again in next week. And occasionally, I'll of course also upload some videos to this channel. So it would be nice if you also watch those. Yeah, and add a thumbs up there. I wish you all the best, and yeah, bye bye. See you next week. All the best.